Everybody. It's nice to see familiar, friendly faces. Welcome to the 2023 Friends Monument Research Symposium. We're so happy to have you here. Thanks for attending with us tonight. I am Colette Straight, Executive Director of the Friends of the Cascade-Siskiyou National Monument. Can you hear me okay, by the way? Give me a thumbs up. Yeah, okay, great. I am here with Zina Brown. She is our new program coordinator. She just started March 13th, and we are so delighted to have her part of the team. She will be acting as the moderator for the symposium tonight, and you can communicate with her via chat, and she'll tell you a little bit more about that in a bit. This is our 8th annual Monument Research Symposium, and tonight we will get to learn about the recent research done in the monument by the 3 university students who received the 2022 Friends Research Fund grants. So thank you so much, Thomas, Hilary Rose, and Heather for being here with us tonight. We really look forward to your presentations. As you may know, the Friends Research Fund annually awards individual grants to undergraduate and graduate students for faculty supervised projects that enhance the understanding, appreciation, preservation, and/or protection of the Cascade-Siskiyou National Monument. Study areas include biology, environmental science and education, sociology, arts and humanities, Indigenous studies, history, and business. So it's really open to a wide range of study areas. Our 2023 grant cycle is currently open, and we are accepting applications until April 21st. We're excited because we doubled the amount of money that we're awarding this year, so folks can apply for grants of up to $3,000 this year, which was doubled from last year, where we offered up to $1,500. So if you or anybody you know is interested, you can find out more information and download the application on our website. Our website is cascadesiskiyou.org. Before we kick off the presentations, I would like to invite our lovely board member, Stassi Maxwell. She's our board secretary, to kick us off by doing a land acknowledgement. So welcome, Stassi. I'm going to mute myself, and I think you can just, I'm going to mute myself and you can just unmute yourself and start speaking.
Thanks, Colette. Welcome, everyone. Really happy that you're all here for our annual symposium. Looking forward to sharing about the lands that we're on. I personally like to do a land acknowledgement that's a little bit more interactive. So I would love to actually ask, and anyone can come off mute, do you know whose lands you're on today, if you're in the Ashland, Oregon area?
Takelma and Shasta.
Yep, that's 2 of them.
There we go.
Thank you. There's one more. What's that, Daniel? Yes. Awesome. So we are on the lands of the Shasta, Takelma, and Lotgawa peoples. Does anyone know where they were forcibly removed to and when they were forcibly removed?
No, well, I was going to say the Takelma.
Yes, Shasta, Takelma, and Lotgawa people. Does anyone know where they were forcibly removed to and when they were forcibly removed?
I think they're all part of the Siletz tribe, correct?
That's one of the reservations they were moved to. Yes, there's another one too. The Confederated Tribes of Grand Ronde. So those are the 2 main places that they were removed to. I do want to highlight, I'm going to put 3 links in chat. The first one is the Confederated Tribes of Grand Ronde do accept donated land. That can kind of fall under the movement of land back, if anyone is familiar with that. And I'll put a link to the Confederated Tribes of Siletz Indians. I wasn't able to find a land back resource for them, but if you're interested in learning more about the tribes, those are their official websites. And I'm also going to put a link in chat to SOU's formal land acknowledgement. as we support, you know, SOU students in their research, there was a, it was about a year-long process for SOU to have a formal land acknowledgement, and I think it's kind of a cool history to share. The Native Studies Department collaborated with representatives from each tribe, and it took a year for them to draft this land acknowledgement. And I also appreciate the work that the Friends do. We do our best to uplift and center Indigenous voices, as well as to make opportunities. Let me go on mute.
Well, thank you so much, Stassi. It's always lovely to hear you speak. Thank you. That was beautiful. So now I'd like to turn it over to Zainab. She is going to introduce our first speaker.
If you wouldn't mind throwing in the chat where you're from and how you heard about the symposium. We would just love to get to know who's with us, and it also helps us kind of figure out how you heard about this. So you can throw that in at any time. Just a few quick housekeeping things. So after each presentation, there will be an opportunity for questions and answers from the speaker. Please remain muted and put your questions in the chat box. And then I will ask the presenter the questions, just so we're not talking over each other. And then please limit the questions to the presentation that was just concluded. And if you have anything outstanding, perhaps we can address them at the very end. And yeah, our first presenter, also we will be recording this, not you all, but like our main screen, the shared screen. So if you have any questions or want to look back on something that was said, we will be posting it to our YouTube channel. So our first presenter will be Thomas Hillard. Thomas Hillard is an undergraduate student in earth science and biology at the University of Oregon. Along with his faculty advisor, Jad Delura, Thomas has mapped out the rock formations in Jenny Creek Falls, as well as a region of tuffaceous rocks, debris flow, and lava around Pilot Rock. So I will turn it over to Thomas.
All right. Audio. I need to be okay. Cool. Need a second to keep the slideshow rolling. Okay, how does this look for everyone? Okay, cool. All right, so hi everyone, my name is Thomas Hillard. Like they said, I'm an undergrad at the University of Oregon studying geology and biology, and I've mapped out a section of volcanic stratigraphy in the Northern California section of the monument with Jad Delura. For the general outline of this presentation, I'll start by talking about like the regional setting, and then kind of move into like the volcanic layers and some other things that we saw in the stratigraphy. We'll talk a bit about petrography, but not a whole lot. And then I want to finish off with the connection to kind of the more biological aspects of the monument and how it ties in, how the geology itself ties into other sections of the monument itself. So for the regional setting, we're talking about the Northern California section of the monument, so kind of close to the Iron Gate Reservoir, right up basically to the border. The volcanic stratigraphy here is kind of the Oligocene age, which, as you can see from the geologic timescale, it's about 33 to 23 million years old. The specific volcanics that we were looking at was about 28 to 25.2 million years old, but I don't think the dates are fully there. I think we're waiting on a few dates to come back in. And this represents Western Cascades volcanism, which was the Cascades essentially during this time period, this was the main mountain range, and then due to a change in subduction angle, the Cascades shifted into the high Cascades that we have today. But these are kind of nowadays they've eroded into the mountains that you kind of have to cross over to get to the high Cascades today, over those passes. And these are the volcanic systems that we're dealing with, at least in the part that we mapped. There's also high Cascades in that area, but that wasn't really the focus of our study. And from our study, we were able to construct a geologic map of the area. The main two formations that we're dealing with are the Colston Formation and the Roxy Formation, which I'll talk about in a bit. The Roxy Formation is younger and is kind of the green parts of the map, whereas most of the other parts that are purple, blue, kind of light brown are the Colston Formation. And you can see the stratigraphy, the layering of the map gets pretty complicated. It mostly goes from the youngest at the bottom up to the top being the oldest, kind of a change in elevation, but it's pretty complicated, and this is typical of volcanic stratigraphy. The kind of splotches of like lava flows and tuffs and airfall like of ash layers and whatnot, and lavas flowing on top of each other. It can be pretty complex, but this is a map we eventually came up with, showing kind of where the different layers are based on what we found in the field, but also aerial image analysis, looking at kind of aerial images of the park and seeing if we can tell where the boundaries of the layers are based on the topography of the region. It's pretty impressive, the amount that you can tell just from aerial images alone, but also being able to go out there and kind of check what you find and mark those layers yourselves. So the two formations that we have are the Colston Formation, which is going to be that older formation, and the Roxy Formation, which is the younger formation. And the main difference between the two is kind of in the content of the volcanic material, with the Colston Formation mostly being volcaniclastic material, that is kind of debris flows, kind of sediments from like volcanic eruptions, volcanic tuffs, which are like ash deposits, and a lot of those debris flows and volcanic tuffs you find in the Colston Formation, but not really in the Roxy Formation, which is that younger, kind of more higher up layer. There are some lava flows in the Colston Formation. The lava flows are basaltic and acidic, which is the kind of chemical composition of the lava flows based on silica content. And that applies for really all the lava flows that you find in this part of the monument. The lava flows are also brecciated and so they're kind of like built up and like blocky in shape, which is again something you see in the Colston and the Roxy Formation. But the main difference is that in the Colston Formation you see a lot of this volcaniclastic material, this like it's more softer, it's more easy to erode, and that affects a lot of things that you see in the park, like topography, like plants and plant life and vegetation, and that stuff that I'll get into later on in the presentation. But the Roxy Formation, in contrast, is almost entirely that lava flow material, that harder, like blocky lava flow that's brecciated, and there's very little volcaniclastic material in this part of the monument. And the reason why that's significant is because this implies some kind of movement of the Cascades during the time between the Colston and the Roxy Formation. Again, you're looking at several million years in between these formations, but a shift from the Colston to the Roxy Formation implies some kind of movement of the vents that apply to these volcanoes. So if you kind of think about the Cascades, kind of north to south, you have a strain of different of where the lava is coming up, and a lot of the times we think about the huge like stratovolcanoes like Mount Shasta, Mount Hood, the Sisters, that kind of stuff. But there's also lots of other little vents and stuff that open that don't necessarily have mountains attached to them, but are still an outlet essentially for all the lava that's coming up from beneath the crust. And the Roxy Formation has a lot more of that lava flow material that you would expect from something that's closer to the vents. And so what we're kind of hypothesizing from this transition as you go from the older volcaniclastic material like ashes and sediments and stuff to the Roxy lava flows is that you have those vents getting closer to this part of the monument. And the main like north-south transect of the Cascades is overall more to the east of or was to the, kind of more to the east of this part of the monument. And so this implies a westward movement of the Cascades as you're getting closer to the vents in this location with more lava flows. And what would cause that? Probably some kind of change to the angle of subduction. As you get a less of an angle of subduction, you get different distances between where that volcano is going to, where the volcanoes will form as you get different distances of melting away. But that's probably what would have caused something like this to happen. And then to get some images for context for what we're looking at with the Colston and Roxy formations. With the Colston formation, here are some images of volcaniclastic material and lava flows. You can see the volcaniclastic material is that white like ash layer, that's that volcaniclastic tuff. And you can see that it's a lot more easily weathered. It's not like a big blocky rock. It's more more like soil almost. And that's going to have a big effect on vegetation and also especially topography as well, because you're not going to build up with that, all those like lava flows that don't erode as easily. You're going to have more flat terrain, which will also affect vegetation as well. And then with the lava flows, you can see that they're kind of blocky. That's that ah shape to them. And they're going to be a lot more cohesive of a rock unit compared to any of the volcaniclastic material that you would see. And with the Colston and with the rock samples, we use petrographic analysis to determine like chemical composition. This is essentially looking through a microscope at a really thin section of the rock to see what kind of minerals and what are the chemical contents of the different rocks units. We'll take hand samples from the field and turn them into these thin sections that we can analyze in a microscope, as you can see in the bottom left. I don't really have the time to get into extreme detail as to what these mean, but basically you can kind of see with the tuff that volcaniclastic material has a lot more shards and kind of glassy material, whereas the lava flow has more cohesive like mineral grains and things building it up, which is going to affect kind of on the macro scale how you see the differences in the volcaniclastic materials versus the stronger lava flow materials, and how that also affects topography and vegetation. The Roxy Formation, again, in contrast, is pretty much entirely volcanic lava flows. As you can see, it's kind of red in color due to weathering, and there's also this nice little rock formation that was kind of poking up out of the ground at the top of a hill. We called that Sphinx Rock. It was pretty cool. But that's the Roxy Formation is pretty much just these larger, chunkier lava flows that you see, again, implying some kind of movement of volcanic vents to support that kind of transition. And again, with the microscope material, you see a lot of the same stuff as you would in the Colston with the lava flows and the minerals and stuff inside of them. And so a couple of other things that we saw just geologically related are, one of them would be unconformities. These are essentially, if the example at the bottom is not from the park, but it's an example of an angular unconformity where you have some layer at the bottom that's older and maybe it got deformed structurally to where you have, it's now at an angle, but then it stopped deposition, it stopped adding new material to it, and over time it began to erode so that you have like a new surface cutting into that layer, and then it starts depositing again, so you have another layer start to build up on top of that. Maybe that gets deformed in a different way so that you have the change kind of in the angle of where it's attaching to. But the main importance of unconformities is that they represent some kind of gap in geologic time. You're not seeing a continuous record, you're seeing some kind of stopping point where there is erosion occurring and you get buildup of more materials on top of that. And with volcanic stratigraphy, you're going to see a lot of unconformities because there's going to be spaces in time in between volcanic deposition where erosion can occur and new layers can occur on top of it, often in very messy ways, as you can see with lavas. And so we've seen plenty of erosional unconformities in the monument. And you can see an example at the top left with part of the Coulson Formation. You can see the tuffaceous material at the very top. That kind of purple row is cutting into the TOCWF unit, while that orange layer, the TOCDC, is kind of supposed to be in between. There's just a lot of mixing of the different layers at that location, and you're not really seeing a continuous transition of layers, but rather you're getting some gaps there as you go up the stratigraphic column. And then another thing that we saw were faults. That's where you have blocks of rock moving against each other. On the geologic map, these are represented by the black transects, the black lines. Most of them, as you can see from the map, trend northwest, which probably has something to do with the larger tectonic setting. And they're mostly pretty steep, up, down, moving like up and down, called normal faults. An example of that is at the top left, where you have kind of a foot wall and a hanging wall, and these are moving up and down towards each other. These weren't the only faults, so there were some lateral faults moving side to side, kind of like what you see in San Andreas, only on a much smaller scale. And how we can tell this there's a couple ways. One of them is slicken sides, which are striations on the rock itself that you can see that form as the rocks move past each other. We've seen many examples of those, and they're pretty cool striations just as the rocks move past each other. And you can also see dramatic changes occur at some locations in between different layers of material. An example of that is in the picture down below, where you can see those vertical formations kind of moved against the layers next to them. And you can see that there's been some kind of displacement there. And so you see a lot of that structural geology, different points in the park, which again just complicates the overall geologic picture that we're looking at here in the monument, and the already complex volcanic stratigraphy. But then finally, another point that I want to mention is kind of connecting the geology that we've analyzed in our study and see how it connects to the monument as a whole, especially with vegetation and thus the entire animal ecosystem. well, there's a pretty direct connection with the vegetation that you can see just by going out and looking at the geology. And a lot of that has to do with groundwater and how water in the soil can move, and how available it is to plants. So a couple things, faults like the ones that you saw on the last slide are kind of opening up these fractures, these cavities, and they become conduits for water that support trees and plants. And I don't have a picture of it, but on the aerial images, you can identify faults just by seeing these lines of trees and dense vegetation that are able to grow because that fault has opened up channels and conduits for the water to grow that support all that dense vegetation. And that's how we've been able to map a lot of those faults. There's also a difference between lava flows and the volcaniclastic material that's present a lot in the Coulston Formation. The chunky kind of cohesive rock units are able to actually fracture more. And because of that, they are a lot more permeable to water flow. And so you end up seeing more plants and trees on those lava flow units than on the volcaniclastic tuff units, because those tuffs are impermeable. And so you don't get you're not able as a plant to get access to a lot of the water that would support the larger plants. And you can see a good picture of this in the image on the left, where you have that that mound, that's that lava flow unit, and you can see all of the shrubs and vegetation just concentrated on that lava flow unit on the top of that hill. But there's a pretty dramatic stop as you get down to the more flat volcaniclastic material, where there aren't any of those larger vegetations growing. And how exactly does that work? Well, the volcaniclastic rocks in the Coulston, they chemically will weather to shrink swell clays in the soil. That's just chemically with the material. What happens when you get a lot of that volcaniclastic and that ash material, you get those shrink swell clays as the soils weather, and those absorb water in the rainy season and then expand and kind of close off gaps that water is able to move through, and so the soil becomes impermeable. And that is kind of the nuts and bolts of how that works. And then you also have the flatter topography of the volcaniclastic rocks, which will also affect vegetation as you get differences in, sun exposure aspect, that kind of stuff, water movement and whatnot. But I just think it's pretty cool that there's such a direct connection to the vegetation and the biological aspect of the park, just from the geology. And we've been able to get a good glimpse of that just by doing our mapping and research of the geology of the volcanic area. And so, yeah, that's about it. Just an overall summary. These are Western Cascade volcanoes in this part of the monument. They date back to the Oligocene, so before the modern Cascades, you have the Western Cascades. The younger Coulston Formation has a lot more of the volcaniclastic materials, whereas the Roxy has the lava flows, implying those vents were moving closer during that time gap. And there's also faults and unconformities present that complicate the geology. And the geology, especially the difference between those volcaniclastic and lava flow materials, will also affect the distribution of plants and soils. And so there is a pretty good connection between the geology and the biology of the monument, which is pretty interesting. But that's about it. Any questions?
In the chat. And Jad, do you have anything that you would like to add? So John would like to know, Thomas, if you can tell us anything about Monument Peak.
I'm not quite sure where that is. Is that in the Northern California section?
Not sure. Oh, he says no.
I'm not quite sure about the rest of the monument. I'm not sure if Jad could chime in on that, but I would assume that if you have a peak of solid material, it has something to do with volcanic lava flows or some volcanic structure would be my guess, but I'm not quite sure what that is.
No, I don't know where it is either, Monument Peak. If you give us some idea of where it is, I might have an idea. But otherwise, there are lots of different places in the monument that really project, such as Hobart Peak and Pilot Rock, and these sorts of things are made up of harder rock units too. In fact, in the picture he's got there, in this very last slide, the question slide, you see some very steep terrain. And that's part of a large intrusion that goes into, that's in Northern California. And you notice that it is very, very resistant to erosion. Most of these intrusions are, and there are a few in the monument in this area.
It looks like John actually meant Pilot Rock, not Monument Peak. So I don't know if you can tell us about the formation of Pilot Rock.
Yeah, Pilot Rock is a small, the remnants of a very small volcano. It's all that's left is really the throat of the volcano or the neck of the volcano. And that which was the lava flows and things such as that are gone. They've been eroded away. That particular feature is 24.5 million years old. So it's about the same age as some of the rocks we're looking at right here in this latter slide, part of the Roxy Formation.
Okay, thank you very much. We do have another question. Do you have any idea how long ago the waterfall on Lower Jenny Creek formed?
Oh, that's something that Thomas and I really didn't get to. I've been there and taken a look at it. The high cascade lava flow that's on the top of it is about 3.4 million years old. So you can do some rough calculations as to the base of that lava flow and the top of Jenny Creek Falls, and you can calculate how much erosion has gone on since that particular 3.4 million year old lava flow was deposited. That's an interesting area because it's about one of the only areas where the high cascade lava flow flowed into a creek and actually produced things that we call pillow lavas. And the reason Jenny Creek Falls is there is because there are a series of dikes, and dikes are like those little intrusions that occur, don't necessarily reach the surface of the earth, but they're very hard. And all the rock around them is soft. So the falls is generated in that manner.
Well, thank you. That was very interesting. If we don't have any more questions, we will move on to our next presenter. Let me do a little bit of zooming here. All right. So our next presenter is Hilary Rose Dawson. Hilary Rose Dawson is a PhD student in biology at the University of Oregon. She has been creating a preliminary list of truffles, which are underground fruiting fungi species in the monument. She is conducting her research with the help of a dog named Rye, and her faculty advisor is Dr. Lucas Silva. Let me let Hilary take over. Okay.
Good evening, everyone, and thank you for that lovely introduction. I'm going to be talking about truffles in the Cascade-Siskiyou National Monument. That was the funded research provided by Friends of Cascade-Siskiyou National Monument. And first, I want to start by introducing the team because this is very much a team effort here. So I'm Hilary Rose. I'm a PhD candidate in the University of Oregon, and my research focuses on plant-soil fungi interactions. My sister, Heather Dawson, is a technician here at UO as well, and she's the one who's trained these dogs and initially got me into truffles and fungi. And of course, the star of the show for us is Rye, a golden retriever who's 2 years old, who's a truffle diversity extraordinaire. What are truffles? So truffles are hypogeous or underground fruiting fungi. If you think about the monument and the summers there particularly, you'll notice that you don't see that many mushrooms above ground when it's dry. Truffles have evolved a way of evading this dry season and also just existing underground year round by folding up and going underground. So in the top row, you can see some truffles that we found that have still that bit of a stipe left over where there used to be a full-on mushroom that's sort of evolutionarily gone underground, but also there's other fungi, other truffles that we find like the bottom row where you no longer see those remnant features. They're fully truffle. And on the bottom right-hand corner, you can see a tuber, which has that intricate marbling, classic of truffles. That's all the convoluted gills that are now underground in this one compact structure. You might have heard of our Oregon culinary truffles. There's the black, the fall and spring whites, and the brown. And those are the ones that have an intense odor that are culinarily valuable. So the black truffles have a fruity, sweet odor. Some people think of them as pineapple-y. Whites have more of a garlicky odor and oddly hints of gasoline, which is much more appetizing than it sounds. And then the browns have a ripe cheese odor that some people like and some people don't. And so when we got started in truffles, these were what we were looking for. Taking a step back, why are truffles important? So most species are ectomycorrhizal. They have a very intimate symbiosis with plants, particularly the trees in Oregon. Many of them are ectomycorrhizal, where the fungi live in the roots of the trees as well as foraging in the soils around the trees. And then they will exchange those nutrients, that water that they collect from the soils in the surrounding area, for the carbon that the plants can fix into sugars primarily through photosynthesis. This is a very simplistic view of what's a very complicated interaction, but truffles are one of those primary interactors for that photosynthesis, helping our trees grow, but also diversity underground in the fungi. Truffles are also a food source for many of our small mammals, particularly if you've heard of the spotted owl here and the redback vole. That's a food source for that spotted owl that's a threatened species in our area. The redback vole and other small mammals are flying squirrels. A large portion of their diet consists of truffles. And finally, biodiversity is important, especially as we face climate change and the change in different like drought, heat, the heat bubble that we experienced in June 2021, things like that are going to be altering our biodiversity. So we need to understand what we're losing and what is changing before it's too late. How did Heather and I get into truffles? Heather's a dog trainer. She loves training dogs. And so she discovered this part of the world where you can combine dog training and all the cool fungi that we're finding out in the Pacific Northwest into this very niche hobby of truffle dog training to find culinary truffles. Primarily, people find them for selling. They are culinarily valuable, so they do sell on the market here, although not as the prices are not anywhere close to as high as European truffles. But it started out as a hobby for us of how can we have fun in the woods with these dogs while also finding cool fungi. What does it look like to be out in the woods with a truffle dog? First, we bring the dog out to the forest. generally off leash because we want the dog to be able to roam into whatever regions it wants to. We're following the dog's nose. Tell the dog truffle, the dog searches intently, and then when the dog digs up a truffle, rewarding copious amounts of whatever reward that dog particularly likes. For Cricket, Heather's first dog, she loves treats, but for Rye, Heather's second dog, the one who's going to feature primarily here, he is totally motivated by a tennis ball. His life revolves around us throwing a ball when he finds a truffle. And here's an example of what that looks like in action when we're finding culinary truffles. So Rye, searching around the base of a tree, he will have pinpointed this tree as a special tree for us, digging it up, following his nose, and Heather figured out what he's looking for. She's going to show him the truffle. Look, there's your black truffle. He's mostly at this point going, where's my ball? He doesn't really care about the truffle itself. He's like, okay, ball now. There we go. And there's his reward. And after he brings back the ball, he willingly gives up the ball because he's mostly just interested in going off and fetching it, bringing it back, and then he'll start all over again. He's off for another truffle so that he can get the ball thrown yet again. And what's really special about Rye is we started him off with these culinary truffles, and he quickly diversified into all sorts of different truffles. Like the Ginea in the lower right corner there, that's a truffle that looks a lot like a clump of dirt when you're just rummaging around in the forest floor, but Rye loves finding Ginea. Apparently they grow everywhere because he's finding them everywhere. And he brings them to our attention, and whether we're out in a commercial forest or out on a hike, he knows that if he finds us a truffle, we will throw him a ball. So this is a map of the truffles that we found in Oregon. And this is one of those things where it looks like truffles grow primarily in the Central Cascades, but no, actually, it's mostly that we hike about a 2-hour radius driving time from Eugene. We're pretty sure there's truffles everywhere, but we need to go out and look for them. One thing that's key here is odors. So I mentioned the odors of the culinary truffles and how we as humans like those odors as part of our food. And the reason that Rye can find these truffles is when truffles are mature, they will produce this odor to attract any sort of animal that will eat them, whether that's insects or small mammals. Because the fungi are underground now, they have lost the ability to disperse their spores through the air like a mushroom might. Thinking of taking a classic mushroom cap and making a spore print, those are spores that the mushroom is releasing into the air. And you can't make a spore print with truffles. They don't release their spores in that way. Instead, they rely on attracting animals to eat them that ingest those spores, move around like animals do, and then feces to defecate to release those spores out into the environment again, spreading the truffles range. And so when we think of culinary truffles, we think of delicious truffles, but Rye can generalize across all sorts of odors, from a more mild truffle with low notes to foul and burnt rubber or rotted onion. These are all truffles that we found during our work at the Cascade-Siskiyou National Monument. It's a huge variety of odors. There's interest growing in how we can harness truffle dogs to find more about truffles. Because truffles are underground, they're very difficult to find if you're just a human without this excellent dog nose. And so how can we harness dogs to do research? How can we learn more about these truffles that are so difficult for us to find? So in the upper left-hand corner, we have an example of a study done in British Columbia with Dexter the Wonder Dog. And they were looking at diversity of what are considered the true truffles, the tubers, although that's just a remnant of how we think about truffles in Europe, where the true truffles are the culinary ones, all tuber in Europe. And then down below, there's a new study that just came out talking about how a truffle dog team went out into the Cambridge Botanic Gardens and found this culinarily valuable truffle growing under a surprising number of different species throughout the year. And that's not something that you would know that was underneath your feet, even in a built-up urban area like a botanic garden in the middle of Cambridge, England, unless you were out there looking with a truffle dog, as well as some science of truffle dogs themselves. And so when we heard about the student grant, we were really excited because this was a chance for Rye to show that truffle dogs can be used for diversity in defining the biodiversity of truffles in a set area. And the monument is particularly interesting because it's an overlap of the Californian and Pacific Northwest flora, which makes it just fascinating, both from a botanical perspective, which is where I'm coming from, but also in these overlap areas, you're likely to find a higher diversity of species of all different types, whether that's birds, fungi, or plants. Once we were awarded the funding, we went out there and did several forays. We've done a foray in fall and winter, and we hope to do one in the spring as well, because truffle diversity is seasonal. In each foray, we identify an area where there's likely to be truffles. We release Rye and tell him, go find the truffles. And every time he digs us up a truffle, we document that truffle with a GPS point identifying what the species was and taking photographs of that truffle. And then we collect specimens for each species that we find on each site so that we can do further analyses. This is primarily DNA at this point because truffles are underdescribed, and we suspect that there's about 370 different species of truffle in the Pacific Northwest, but a lot of those species were described using analogies in Europe. So we're also, a lot of researchers that work in truffles are working on redescribing those to be their own unique North American species. You don't need to know the details here, just know that we're taking the sporocarps, the actual truffles, taking dried tissue from them, using a solution to extract the DNA from that, running it through PCR processes, and then using a new technique called PacBio to learn what DNA there is in there in multiple different regions so that we can cross-reference those regions with international databases like NCBI's GenBank Blast. But we don't rely just on molecular. I'm a bit of a natural historian of the old school, and it's just, since we're working with undescribed species, we can't necessarily say, oh, the DNA knows what's up. We also take spore pictures for each of these truffles and take close photographs that you'll see here showing what this truffle looks like when it's fresh and comparing the measurements on those spores with the known description of what is coming out of our DNA analyses. So what did we find? One of the sites that we prioritized in the fall was high elevation mixed conifer forest. And we went to 3 different sites and we found 6 genera and roughly 12 species. We're not sure exactly how many species because we haven't got our DNA evidence back yet. And even then, there will be a little bit of untangling to be done. So here, what we're looking at is lots of different colors and shapes of truffles, lots of different odors that I can't convey through a slideshow. But we're also looking at some things that are multiple species within a single genus. So in the yellow box are our leucogasters, Leucogaster citrinus and Leucogaster rubescens. Both of those smell like very sweet artificial banana. And they were growing in similar habitats, but you can tell from the peridium, the outside, that color is quite different from the rust red in the rubescens and the yellow in the citrinus. So 2 different species there. The red box is a Gaudiuria, or rather 2 Gaudiurias that we suspect are different species, but right now Gaudiuria is undergoing major taxonomic redescription. There's only a handful of species described in the Pacific Northwest, and we suspect there's many, many more than that. And then finally, on the bottom are our tubers, which are glorious in their array of different colors and shapes and sizes. Most of the odors are similar, but they'll have different notes if you pay attention. And the marbling on tubers is just that classic attractive truffle appearance. Another one is in between the red box and the green box, there's the bright white truffle with the conifers in the background. That's Quaramyces alveolatus, which is a species of interest for rare fungi in Oregon. And this one's probably not as rare as initially thought. Some of the other Quariermyces are indeed very, very rare here, but it's interesting that this truffle is in the monument. Here's an example of what it looks like to go on a foray with rye. The white line here is where I was carrying a GPS unit while following rye around the forest. And so that's how we're walking. And you can see that he's going from truffle to truffle. He's not necessarily following a lot of dead ends. The individual dots represent individual truffles that we found, and the different colors are the different genera in this one field site. And from this, you can also see that the truffles are growing in clumps. We can't necessarily say that this is one individual, but it is interesting to note that even within a reasonably small field site, that's a cluster of that genus within that field site. While we were out there in the fall, we also visited a mixed oak conifer woodland site where we found only 2 species of 2 genera, Elaphomyces and Ginea. Both of those are something that we consider quite weedy from just our experience truffling in Oregon. And we think that the diversity at that point was still pretty low, primarily because these forests were still dry. We did this foray about a week after the first rains in the fall, and that's not enough time for these truffles, especially baked in the lower elevation oak areas, to really develop. But when we came back to the oaks in February, after lots of rain, we found a good variety of different truffles. We visited again about 3 different sites, and we found our Elaphomyces and our Ginea again, but we also found other species that we hadn't found previously, including a Zalaramyces that we didn't find at any sites. And in the lower right-hand corner, we found a Balsamia, which has an interesting story behind it. This Balsamia was growing absolutely everywhere in our sites, but we started noticing a pattern. Rye would only find them at the base of a ceanothus shrub, which is known for being a nitrogen-fixing shrub that partners with a bacteria, and an oak tree, which there's a lot of the monument, if you're looking for oak habitats, that meets this description. Plenty of ceanothus growing with oak. But you can see in the map here that we would keep trying to get rye to go back into the oaks where there wasn't ceanothus, which was growing simply around the edges, and he wasn't finding anything. He would instead turn around and say, no, the truffle's over there next to the Balsamia. And so we have this oak that's ectomycorrhizal, a Balsamia that's an ectomycorrhizal fungus, and a ceanothus that's arbuscular mycorrhizal, so totally different way of relating to its fungi, but it was fixing nitrogen with this bacteria. And we suspect there's a nitrogen story here. Maybe the Balsamia needs a certain level of nitrogen to fruit, or maybe the oak roots are seeking out these nitrogen-rich areas. We don't know. It's probably going to take something like stable isotopic analysis to find out more, but this is just a really exciting pattern to be finding even a few forays at a site. In conclusion, we found lots of diversity, and rye was super successful as a method of finding truffle diversity in this monument. We found what we're guessing are approximately 20 different species, just in 2 forays in 3 different habitat sites. Every site that we went to, we found truffles. And that's really cool because even up here, I'm based in Eugene, Oregon, where it's quite moist and there's lots of Doug fir. So there's lots of truffles up here too, but we don't find truffles exactly everywhere we go. And here in the monument, we were finding lots of truffles and they weren't all just one species. So that's super exciting. We're still waiting to get back our DNA analyses from these first 2 forays. And once we have those analyses, I think we're going to be able to say a lot more certainly what we were finding and how it's relating with its environment. We suspect we have some undescribed species. We suspect that we have some specimens that we can't necessarily identify with just the sporocarp, the dried truffle that we still have. We do honestly need those DNA analyses. And we still hope to come back down for a spring foray, tentatively in May, to learn more about what's showing up in this 3rd season. Once we're done with all of that and completing the DNA analyses for our spring foray as well, we're going to write up a list of known truffles of the Cascade-Siskiyou National Monument as a springboard for future fungal and truffle research in the area. None of this would be possible without our advisors and mentors, Lucas Silva and Biddy Roy, who've both been amazing in supporting our efforts and making sure that we have the lab space and lab time that we need. Scott Loring and Jonathan Frank, both amazing fungal researchers who have helped us identify the best spots to be going to find these truffles. And then credit to my sister, Heather Dawson, for all the truffle pictures, as well as many of the beautiful photos and, of course, the cute dog pictures. And a huge thanks to the Friends of the Cascade-Siskiyou National Monument for making this research possible. We've had so much fun researching these truffles down there and exploring this gorgeous part of the world that we otherwise wouldn't have visited. So thank you.
Have any questions, they can throw those in the chat. No questions? Give it another minute, maybe someone's typing. Oh, thank you. It was, yeah, someone just wants to say thank you. Very cool project. Tula is wondering, will a truffle species be named after Roxy?
If you mean the dog Rye, it would be interesting to name something after Rye because his name is also a botanically important species because we eat rye. And so it might be a bit confusing to name a truffle that might grow under, say, an oak or a Doug fir, lolium or rye, essentially, but it would be fitting, I agree.
And someone else wants to know if the different scents of truffles are attuned to different species or different organisms. My bad.
That's a great question. There's a wealth of research to be done on the biochemistry of these odors. It's so interesting to think about the fact that rye can generate these generalize across these odors, which we as humans smell as totally different things. And so it's difficult to say which odors are attracting which organisms. We do know that some truffles are more likely to be eaten by insects versus some more likely to be eaten by like squirrels, but we're not entirely sure why, if that's a placement factor or a different scent or just opportunistic. So great question. Hopefully we'll have future research on that. If anyone knows biochemistry, please.
Okay, well, that looks like the last of the questions. Thank you so much again, Hilary. We are going to finish up the symposium with Heather. So Heather Stewart-Ahn, I hope I pronounced that correctly, is a master's student in biology at Eastern Washington University. She is studying a rare and vividly red lichen. I'm going to try to say its Latin name, Umbilicaria phaea coccinea. She can correct me. I'm sure she will. That is found in the monument. Her faculty advisor is Dr. Jason Ashley at Eastern Washington University, and she is primarily working with Dr. Toby Sprybiel at the University of Alberta. Heather, please take it away.
Sorry, I didn't unmute before I went into share screen and then the button was gone. Okay, this is better. No, go away. All right, sorry about that. Can everyone hear me okay now? I hope so. Good evening. As Zeneb already mentioned, my name is Heather Stewart-Ahn. I'm an MSc candidate at Eastern Washington University. Before I begin, I want to thank Friends of Cascade-Siskiyou National Monument for organizing this event and for offering their generous research grant awards. Also, I'd like to thank Thomas and Hilary for sharing their work tonight. And I hope you enjoyed this presentation. Thanks to those who are attending. My presentation is titled Surveys of Umbilicaria phaea var coccinea, a rare lichen endemic to the Western United States. It's worth noting, unfortunately, misconceptions about lichens are prevalent. For example, a simple Google search definition of a lichen states incorrectly that they're plant-like organisms, absent of mentioning the fact that they're primarily fungal, and more importantly, that they're symbiotic organisms. In fact, lichens served as the impetus in formulating the concept of symbiosis during the late 1800s. Hence, contrary to what Google asserts, a lichen is a symbiotic organism featuring one, a fungal partner known as the mycobiont, which serves as the taxonomical basis, and it also provides the bulk of the lichen body, and nearly all of these are ascomycetes. Two, a photosynthesizing partner known as the photobiont, usually green algae, and in some cases cyanobacterium. Among roughly 20,000 known lichen species, about 2% are tripartite, which means they feature two photobionts. In sum, lichens as symbiotic organisms can be very complex. Generally speaking, there's three different growth forms for lichens, categories of lichens. Firstly, there's microlichens, which are more two-dimensional in appearance, and they adhere very closely to their substrate. Next, we have macrolichens, which can have a range of growth forms, such as hair lichens, foliose lichens, and fruticose lichens, which are radially symmetrical. Lastly, there's a third category that appear as little granules and essentially look like a coating of dust. These are called leprous lichens. Using the species Litharia columbiana as an example, here is an overview of basic macrolichen anatomy. A lichen body is known as a thallus or thalli, plural. The exterior is known as the cortex, which is a hydrophilic, meaning water-loving, outer seal featuring high-density fungi. Additionally, in recent years, new research has demonstrated in many lineages of macrolichens, there are other obligate symbionts present, such as different yeasts, as well as bacteria. Next, we have the medulla. This is the hydrophobic interior, meaning it is more repellent towards water. And it's constituted mostly of low-density hyphae, that would be filamentous threads of fungi, which make contact with this other layer known as the photobiont layer. You might be able to see it as a hint of green lining the interior of the cortex. And in this case, it's green because this is a green algal lichen. Lichens are important for many different reasons. They're capable of occurring in nearly every ecosystem on Earth, including places that are inhospitable to most life forms, such as the Arctic. They account for 8% of the land coverage biomass. They facilitate water retention in their ecosystems. Since they're non-vascular, meaning they directly absorb everything in their environment, whether it be water, nutrients, as well as pollutants. Therefore, they've been utilized worldwide as effective bioindicators for air pollution monitoring. They're also very vulnerable yet resilient. You can easily tear apart a lichen with your bare hands, yet one species survived 18 months of exposure to raw outer space by a study in a study by the International Space Station. Consistent overall with the kingdom of fungi, lichens are very, very talented chemists, and they produce countless secondary metabolites. These are typically compounds that serve defensive roles to prevent herbivory and other things such as UV protection. They also provide a lot of food and habitat for a range of organisms. They contribute to converting carbon dioxide in some species, particularly the cyanolichens, fixed nitrogen. Due to their slow growth rate, lichens can also be utilized for a procedure known as lichenometry, in which the size of a lichen is used to date the age of a glacier. And lastly, on a personal note, for me, they teach me a lot about beauty, reverence, stillness, and more. The genus Umbilicaria is a foliose, meaning again, leaf-like lichen with a distinct upper and lower surface, and it's exclusively saxicolous, meaning it dwells on rocks. Another defining feature of this genus is that they're umbilicate. They have a central attachment point on their lower surface that can kind of be seen on the upper surface as a bulge or these little knots. Here you have what might look like two different species, when in fact they're actually the same species. It's just different varieties of this species. They're indistinct taxonomically according to DNA barcoding of the fungal partner. In brown, you have Umbilicaria phaea var phaea, and in red, you have Umbilicaria phaea var coccinea, which is currently categorized by the IUCN Red List as vulnerable, just one tier below endangered status. A study published last year comparing the chemistry of these two varieties was able to detect an anthraquinone, a pigment that was present only in the red variety. Nonetheless, these remain considered to be the same species but different varieties. While these two varieties co-occur, as you can clearly see here, the brown phaea var phaea is common and widely distributed, whereas phaea var coccinea has an extremely limited distribution, heavily concentrated around the Oregon and California border, within and around the Cascade-Siskiyou National Monument. In addition, it has some very, very striking disjunct populations, such as here in the Ochoco National Forest and two in central Washington. I was able to access the site location data by the authors of the study that detected the anthraquinone pigment because it's no longer publicly available due to its vulnerable red list status. And in addition to utilizing that data, my surveys were greatly enhanced by help from some locals, Steve Sheehai of Klamath Falls, Oregon, and Zane Walker of Copco, California. I actually met Zane on iNaturalist, and he is an undergrad student at Oregon State University, working with one of my colleagues, Dr. Bruce McCune. Zane ended up accompanying me and helping me with some of my surveys. Now here, instead of the previous map that just merely showed the location data, until now, no one has endeavored to inventory these sites and determine the extent of these populations. How many of these lichens are actually present at each site? And after visiting 44 sites, 25 of which were confirmed, then additionally, I went to 19 randomized points that were generated by 5-mile buffer zones created based off of the confirmed locations. I ended up the biggest takeaway of my surveys is that the large abundant sites are actually very, very rare, whereas typically when this lichen occurs, it's very sparse. As you can see, 1 to 3 thalli will be present at a site. And also it's worth noting to give this caveat that of those 25 confirmed sites, in some cases, I actually fused multiple sites into one site because there were so few lichens present. For example, you can't see maybe the best here on the computer screen, but one spot that was within the monument along the Lone Pilot Trail was originally considered 7 different locations. And after I surveyed, I consolidated it into just one site that had roughly 10 thalli total present. Another thing to keep in mind is among the official record known sites, 17 of them, it was extirpated at 3 sites, 2 due to wildfire and 1 due to wildlife activity. And then just moreover, to keep in mind that of all the sites I visited, 57% of these sites had less than 100 thalli. So it's possible, although I'm not completely familiar with the criteria process for the IUCN Red List, that this lichen following this survey work might qualify more as an endangered species variety rather than merely vulnerable. And then moving on, here's some examples of what these sites look like. These are two of some of the most abundant sites here in Copco, California, at what's known as Horseshoe Ranch. This is a natural wildlife area operated by both the state of California as well as the Cascade-Siskiyou National Monument. And this is one of those disjunct sites in Vantage, Washington. And as you can see, the most abundant sites are always very exposed. There's pretty much no canopy cover. And they're in proximity to major waterways. In that concentrated distribution zone of the California and Oregon border, this lichen absolutely has an association with the Klamath River as well as the Shasta River. And then in Washington, it's very close to the Columbia River. And then additionally, there always needs to be profuse basalt at these sites because that's the only substrate it will grow on. Technically, it's considered to be present on ultramafic rock. And I found it on like a amalgam of serpentine and basalt at only one location that was really unique, where the Siskiyous were sort of colliding into the Cascade Mountains right off of I-5 near the Shasta River. And then another thing that I noticed that's kind of worth noting is this huge site at Horseshoe Ranch, as well as the huge site in Vantage, Washington, and other of the more larger sites, they're also all near very large reservoirs. They're not only near major waterways, but these are dammed waterways, which might play a factor in its distribution. I'm not sure about that yet. Here is also more views of just some of the more abundant populations at Copco, California and Horseshoe Ranch. And then, for example, though, at some of these survey sites, I would just find one little red lichen like that amid all of the brown phaeas. And overall, I was also taking measurements of the thallus sizes, and they can range from as small as 2 millimeters wide to 38 millimeters wide. And on average, I'd say the typical size of one of these lichens is about 7 millimeters. So they're very small. I actually did not realize how small this lichen was until I saw it in person. From photos, I thought it was actually much bigger. And here on the right, this Creek Falls site, this is one of the sites that Zane took me to very close to the Horseshoe Ranch site near Copco, California as well. And this site was the most abundant site. And if there is somehow a mother site that begat all the other populations, I have a suspicion that this could be it. And here's some more examples, though, that it's actually just very, very sparse. Sometimes I don't really find hardly any of it at all. This was, again, the population I mentioned that was originally 7 sites off the Lone Pilot Trail in the monument that I fused into one site. This is a roadside site near Keno, Oregon, and it had very low abundance, yet this one little speck that you might be able to see towards the top of the boulder ended up being one of the largest thalli that I observed. And then another thing is at the bottom, this is one of the new sites I discovered from those randomized points. I neglected to mention the purpose of those randomized points was to generate absence data to help construct a species distribution model, because you need to know not only where this organism occurs, but also where it doesn't, to try to determine what influences where it exists. And I went to one of my randomized points. It was roughly halfway between Ashland and Klamath Falls, just outside the monument along Highway 66 in an area called Camp 4 Creek. And I went to my randomized point. It was heavily forested and there were no rocks. However, as I was leaving and I was going down just slightly up the road from the randomized point site, I saw this wall of boulders and I thought, there's rock, I should stop and look for it. And sure enough, I did find some, but it was only 3 thalli for that whole entire rock pile. So unintentionally, some of my randomized points, which were supposed to illustrate absence data, I ended up generating data for new sites, but none of those new sites were ever these abundant mega sites that have over 300 thalli at them. In addition to the fact that no one's ever inventoried this species variety. Part of what prompted me to do research on this particular lichen is that in August of 2021, I visited the Horseshoe Ranch site just as a luxury lichenizing adventure because I was in the area doing other field work. I'd never seen it in person. I really wanted to go see it. And I ended up encountering at the Kopko site here in the upper left and the upper middle photo, an unprecedented lichen specimen. It featured both varieties on the same thallus. I don't know if you can see, but there's just a little portion on the bottom that's brown, whereas the rest of the lichen is red. And this raises a lot of questions about, once again, the complexity of lichens as a symbiotic organism and whether or not it's possible, also given the nature of the complexity of fungal reproduction, which can involve steps where there's more than one nuclei within a cell during the reproductive process, can there be intrathallus genetic variation within a single lichen body? In other words, can two different fungal species or varieties appear on the same thallus? And clearly they're doing it here. And so as part of my survey work, I was also trying to determine how common is this occurrence of these chimeric thalli. And it's not very common from all the scouring that I did. And I only found one more at the top right near the Shasta River. And that one was actually one of the strangest ones out of all these chimeras. You might not be able to see very clearly in the smaller photo, but the red portion is clearly like more raised than the brown portion. And it was very mottled. It's a very, very strange chimera. And then the bottom three photos are all specimens that I collected from the Vantage Washington site. So that's another thing to consider is like, for some reason in particular, the Vantage site is producing more of these chimeric thalli than any of the other sites. And again, like I said, this is an unprecedented thing in lichenology. There's only one other instance of someone making a similar observation in 1978 with a different genus, Haematoma, in England. So in conclusion, for future studies, originally, actually, this project was going to be my main thesis, and I was going to pursue some of these other research objectives. And I would love it if someone picked up the mantle on this. It would be wonderful to complete more inventories of additional sites, to do comparative genomics, to determine are there nuances genetically between the two varieties, that DNA barcoding, which focuses on a limited portion of the DNA for speciation. Maybe there's other things going on in its genome that generate great differences between the two varieties. The component that I was most excited to try to take on would be population genetics, comparing the genetic trends of the different populations to determine how closely related are they, especially the disjuncts, those really, really isolated, far remote sites in Washington versus the ones in Oregon and California. Is that convergent evolution? They were subject to the same environmental pressures that caused them to generate the red pigment, or did they somehow disperse all the way up there? And then lastly, for a future study, to construct a species distribution model to determine what factors influences where the Umbilicaria coccinea occurs. In addition, this future studies research objectives would help in answering the question that no one's been able to answer yet, which is why is coccinea red? And four potential hypotheses for the cause of this red pigmentation is one, transcription, meaning do both of the varieties have this gene for producing the red pigment, but a certain condition or maybe even just random circumstance prompts that gene to activate and produce the red pigment. And then that's also related to the second hypothesis of ecological adaptation. It's already been demonstrated in one other species of lichen that's very closely related to this one, Lasallia pistulata, that it will have a different pigmentation depending on how cold or warm of a climate it's occurring in. And then thirdly, another possible scenario is that those auxiliary symbionts I mentioned in the overview of anatomy, such as the yeasts and bacteria, these could actually be responsible for the red pigmentation and not necessarily something that's occurring from the fungal partner. And then fourthly, in my opinion, after all the surveying I did, considering that there seemed to be no rhyme or reason to those sites where you would just find one amid a bunch of brown ones, it could actually just be a random genetic mutation. Or another scenario is that it was an ecological adaptation, that this red pigmentation is prompted by a certain climate factor, but that original population that was responding to climate somehow just proliferated and reached other areas. So thank you to Friends of Cascade-Siskiyou National Monument again, the American Bryological and Lichenological Society, Zane Walker, Steve Sheehai, John Villela, Devlin Madrone, Bubba Pfeffer, Dr. Bruce McKeown, Dr. Jason Ashley, and Dr. Toby Shprabilla. Thank you for your time. Any questions?
If anyone has any questions, please put them in the chat. Hilary says, fascinating questions about fungi, lichens, and symbioses. You mentioned that this lichen has a pigment that the other Umbilicaria doesn't have, and yet it isn't considered a separate species. What evidence would convince taxonomists to separate them? Does the presence of chimeras make it harder or easier to split the species?
That's a lot. Those are many different questions. I'll start with the last one first. You know, it depends on the perspective. So when you have these species varieties that are based off of chemistry, in this case, the red versus the brown, those are also what's known as chemotypes. In the observation I mentioned with the other species where someone did encounter a dual thallus of two chemotypes on one thallus, their conclusion was that this illustrates that they're the same species. But overall, this is where the comparative genomics could elucidate more information about our conceptions of speciation in lichens and fungi that without trying to get too technical and complicated, like I said, there's a specific region, what's known as a loci of DNA that's targeted for speciation of certain organisms. And in the case of fungi, it's what's known as the ITS region. But genomics used to be less accessible and much more expensive. It's still fairly expensive though. Through more and more genomics work, we might be able to find other indications of speciation that the traditional loci that we use isn't able to demonstrate. I could be mistaken about that, but I also just definitely believe that things are always much more complicated than we allow them to be. So I think I tried to answer most of those questions. If I missed something, would you let me know, Steph?
Oh, she says that's great. Okay, wonderful. Any other questions? Well, thank you, Heather and Thomas and Hilary, for your wonderful presentations on your research topics. I am going to give the floor back to Colette.
Sorry to interrupt. I also just wanted to mention to the people that are in the Ashland and Cascade-Siskiyou National Monument area that I'll actually be hosting a lichen walk on Sunday, June 25th, and we'll be going to that Horseshoe Ranch site. And it's actually really one of the most spectacular sites of this lichen, and I encourage people to attend. It'll be free. It's a fairly easy walk for able-bodied people. It's about 1/2 to 3/4 of a mile, relatively flat.
Yes, that will be one of our Sunday hikes coming up, and there will be an opportunity to sign up for that soon. Colette might have a little bit more to say about that. Just
Picture share. Well, hi everybody. Thank you again for those amazing presentations. I learned so much and it was so impressive. Thank you. Let's see if I can make this larger. For this guy? No, this guy. Can you all see that? Okay, there we go. Well, shoot. Yeah. Oh, there we go. Okay, great. Anyway, thank you all again for attending and thank you for your questions and your attention. Before we say goodbye, I just wanted to let you know about our upcoming events as displayed here. We have our Hike and Learns scheduled out through June so far. Hike and Learns are a Friday evening talk followed by a Saturday hike in the monument. So on April 28th and 29th, we have oak trees in the monument with Rich Fairbanks. On May 12th and 13th, we have history of the 1846 and to 1860s Applegate Trail with Jeff Leland. On May 26th and 27th, we have wildflower identification with Liz Landreth and me. And then we have June 9th and 10th, monument geology with Jad Delura. We have some stewardship days scheduled on May 20th, the fence pulling with the BLM out of the Green Springs Loop Trail. And June 7th with KS Wild, we have a star thistle pull at the monument or at the Mariposa Preserve. Finally, we have our Sunday hikes. The first one will be May 21st to the Buck Rock Tunnel. It's a history hike with Chelsea Rose and SOU's Laboratory of Anthropology. It's going to be in honor of Asian American and Pacific Islander Heritage Month. And then June 25th, like Heather said, she's going to take us out to Horseshoe Ranch to look at the samples of the red lichen she was just talking about. We're going to also, probably have another speaker there to talk about the Horseshoe Ranch wildlife area in general. So it's part of a monument that like most people don't go to. And then we have two other wildflower hikes, July 2nd and July 16th, also with Liz Landreth. So you can sign up for those free events on our website at Cascade-Siskiyou.org. Let's see. So yeah, if you're not already signed up for our email list, you can do so by going to our website and subscribing. That's a great way to keep track of what we've got going on because some of these events aren't available. You can't register for them yet. So, and then we'll be adding additional events through this like 2023 programming season. So if you get on our email list, you can be made aware of like all the cool things we've got going on. Yeah, so of course, we'd love to see you at those events. And it's so nice to see all of you now. We offer all of our programs at no charge to participants. And so we are funded solely by donations and grants. So thanks to those of you who have already donated to us and have donated to us many times in the past. And we invite you to continue to donate. And those of you who haven't donated to do so by going to our website. It helps continue our work in support of biodiversity in the monument. Unless we have any further questions. Yes, here's a question from Jonathan. Can you share if this is recorded? Yes, this is recorded. We will put it on our YouTube channel and also in our next newsletter and on our website, we'll have places that we'll have the link so you can watch it back. Yeah, any other questions before we say goodnight? Just give it a couple more seconds. Yeah, you're welcome. And thank you. Thank you again to the researchers and students and presenters and all their supporters and to all of you. It's nice to see you. Good night.